Let's start with Amaya Álvarez, that she's also one of the responsible of this forum, on the organization of this forum. I want to thank Amaya for her help on organizing the forum once again. She's a research technician at the Fish Ecology Group of the IMEDEA of the CSIC and Universidad de Illes Balears, in the Illes Balears here in Spain. And she's going to present the automatic operational and high-resolution monitoring of fish length and catch number for on landings using deep learning. So, Amaya, thank you, and go ahead.
Hola.
Ah.
Hola, justo
Hi, Amaya.
It just went out. I don't know why. Sorry, I'm here again.
Okay.
I will share. I had just shared the screen. Okay, let's go. I don't know. One moment. Sorry.
Yes, no problem.
Now, this one. Now I think you can see it. If I put like this, can you see the presentation?
We are seeing the PowerPoint framework, not the presentation.
Okay, I don't know why, because if I put okay, well, sorry, again, I will do it again. I do not, well, if I put all the screen, you will see, no?
Yeah.
Okay, so I will do it like that. I don't know why it doesn't allow me to share the full screen, so I will have to do it like the
with the PowerPoint is okay,
with
no
the
problem.
PowerPoint. Okay, so what a pity. Okay. Well, hi, I'm Amaya Álvarez, and I'm from the Fish Ecology Group at IMEDEA. I'm a PhD in marine science, and the name of this presentation is the title of the last article we have submitted related with deep learning. But I'm going to talk mainly about the 4 years that we have been working with deep learning related to fish identification in images. So the Fish Ecology Group at the IMEDEA institution, it's placed in Mallorca Island in Spain, in Mediterranean Sea. It's a research group that it's mainly committed to the development of new tools to infer population dynamics, as well as just studying the multi-scale connections between environmental changes, fisheries, and impacts on society. Well, this group is mainly formed by biologists, so we don't have too many informatics or physicians or engineers. And the group has been recording lots and lots of video and taking lots of photo, mainly underwater, but also like in this presentation on the auction, on the fish market. So here you can see more or less the images we are working with, and the methodology we have been using for the last few, well, till the last, I don't know, 4 years, 5 years, was mainly the visual analysis, which is really time consuming. So since 2018, there have been various projects in which deep learning has been introduced as an analysis tool. Well, I'm presenting here 4 of which these projects, but in the group there has been other projects related to deep learning, and where the objectives were not the fishes, but the sea plants or the boats, for example. In 2018, we began with PhotoPage, which was at the beginning was not thought to be done with deep learning, but due to the poor results that we were seeing at the bibliography, we decided to ask to the computer vision department of the University of the Balearic Islands. And so we decided to try the deep learning methodology to see if it worked fine with the fishes. Even after this photo page, then came photo page 2 and Retorno, which were all projects funded by the Fundación Biodiversidad. And all these projects were trying to automate processes that take a lot of are like really time consuming for people that has to visualize all the images, to search the fishes, to measure the fishes in the images, and so long. So we had these 3-year projects, one after the other, and always in collaboration, but in external collaboration with people from the university, from the computer vision department. And finally, in 2020, we get this project in collaboration with this TAMI group, with this group of treatment and mathematical analysis of images. And now we are doing this project, which is with underwater images. So I'm not going to talk about this project at the presentation. Well, we work with images that come from the fish market of Palma de Mallorca. And these images have been daily available since 2019. But before that, in 2018, the images that we got were taken by us in random days during the auction. So we didn't, in 2018, we didn't have a lot of images. Then after, in 2020, the video, well, we get after 2020, we get daily images with metadata associated that came with the weight of the box, the species, and the boat of where the fish come from. And what we want to extract from these images are the number of the fishes, the number of fish that are inside the box, the length of the fish, and the species classification if possible. This species classification even now is get by the metadata. This is not the case for all the fish markets. And even more, you can see here in the middle, there is a mixed box, which is usual here in Mallorca, having this kind of boxes where are different species inside. We have worked with dolphin fish, with Zeus Faber, which in English I think is John Dory, and with Mullus barbatus and Mullus surmuletus, which is red mullet, and as well with the hake. All these species were selected either because of the good placement in the box, which seemed to make it easier to work with, or because of the economical value of the species. We have worked with we have tested 3 different kind of neural network, but we have mainly worked with master CNN. We have used it for the hake and for the dolphin fish. We also try to use RetinaNet for trying to avoid the complete segmentation of the fishes because it's really, again, time consuming, having this, making all the training data set. So we try to use RetinaNet, which is only bounding boxes. It didn't give us really good results. I think maybe because of the small size of the training data set, it could be, and also because the difficulty that we had to make differences between the Mullus surmuletus and the Mullus barbatus, which are really similar and are difficult to visually make the difference and label all the images. We also use the VGG16 just to make a difference in between red and blue fishes to extract the images we wanted for creating the the training data set. But what we have used most is the masker CNN net. I have to point out that we start using this neural network because the way it was shown in GitHub make it really easy to do the implementation. Again, we are like mainly biologists, and we didn't have at this time in 2018 the background in programming in Python and using the GPUs, etc. So this the Matterport implementation that was at GitHub gave us like really easy to implement the network and to make all the trainings with our own data set. For the trainings we use, for making the data set, we use the label box first annotation software, and then in 2020 we changed to Supervisely. We have segmented 2,600 heads of hake and more than 4,000 dolphin fish. The hake and the dolphin fish were used in different studies. The hake was a study done in 2018 and 2019. Here is the article that was published in an special issue, where we tried to infer the total length of the hake, just segmenting the heads. So using the head in finding the majority of the heads inside the box, we try to infer the total length. We get really good results, and mostly in the part of the deep learning, which gave us like really good results segmenting the heads and having like few false positives. Then we make the study of the Yampuga, and what we wanted to show in this study, which is now we can use the title of this presentation. What we wanted to show here was an operational system that uses deep learning coupled with a statistical model. Here is this small plot is a deep learning was used to extract all the number of Yampugas. So what we've done is estimating the number and the mean fork length of the dolphin fish caught in the Mediterranean fishery. So this system operates with these images of fish boxes automatically collected at the fish auction. And with a statistical model, we correct the biases due to detect fishing using the convolutional network. And we estimate the mean fork length for each box. So here you can see at this slide on the left hand, you can see how the process goes from the boxes going into the fish auction, capturing the image, and then coming into the institute to make this analysis by deep learning and the statistical model, and then get the mean fork length associated to each box. Here at the right top panel, you can see how for the last season, for the season of 2020, how the number of, sorry, sorry, here at the top, we can see the length, how the length is growing through the season. So the mean length for each box, as well as the individual length. And on the lower panels, you can see how the number of fishes decreases along the season, as well as the weight of the total count of each day. So as the season goes on from August to December, the fishes, the dolphin fish is growing, and the number of fishes caught are decreasing. And here I show you the matrix confusion for the max CNN we use. I can notice that you cannot see the numbers. for the mass scale CNN, we got a mean average precision of 84%, which is not really, it's not, we should have a better results. We try doing different tests, but we couldn't get, like, we don't know which one was a problem. Maybe the size of the fish inside the box, or the number of images were not enough for the training. Maybe the GPU, we didn't have a good big enough GPUs, because we tried many different ways of training, and we couldn't afford better results. So what we have learned at the end of all these processes about deep learning in fish auction images is that it's really important to select the images for this small training data set. As I'm showing here some kind of images of dolphin fish, which are, the first one here, the dolphin fish is ordered in a different way of the normal ones, which are placed like this. But in this case, like our really small ones are placed on the other side. And also, it's need to be the segmentation to really be well done. Here you can see that the tails are not well caught by the segmentation. You only get the body. And so the necessity of understanding well the image segmentation. We have used this technique for the heck, and it worked really well. But we tried to use with the dolphin fish, it seemed the same problem. It didn't work at all. We tried different ways of using it, and we don't understand why, but it didn't work. And as well, we understand that these kind of problems, we need bigger training data sets. It seems like having 4,000 fish are not enough for giving really good results. I think this is all. I don't know if you have heard me. It's really weird talking like that. And that's it. Thank you very much.
Okay.
SICAPTOR, monitorización electrónica para una mejor gestión.